Okay, so good morning, lunch and evening. Very interesting to hold this sort of global talk. I've never done that. So thank you for being invited to talk about the area where we work in my group. And we've been working with gene editing in Salmon for the last 10 years. And now I've been happy then to be able to look further into all other groups that have been working with this technology in fish for the last 10 years. And it's really a lot of interesting cases I have to go through. So the talk will first consist of trying to share the current situation for using gene editing in aquaculture and the specificity of fish. And then I end the talk a little bit about when we've been going into the regulatory landscape, just to be able to do some research trial here on our facility in Bergen, Norway. So first of all, aquaculture is a really growing production of fish. It has just surpassed the capture. Wild capture in amount, so it's really exploding, and there are about 30,000 species of salmon or fish, and only then a few are in aquaculture. So the opportunity are large. And if you look at this overview then from FAO, you can see that most of those aquaculture fish are then farmed in freshwater and not so much seawater, where you have the most wild capture. And then there is also quite a lot of aquaculture of lower trophic levels, which I'm not going to go into in this talk, but there's really some interesting cases here also for gene editing, and that has so that those are the technology is moving into those too. So when you think about that, there are about 30,000 species of fish, and these are the main ones that are used in aquaculture. You see there's just a handful of fish that are used for aquaculture. However, technology is moving forward and breeding genetics, and so it's very interesting to be able to then use this gene editing technology to maybe improve or make the aquaculture of these fish more sustainable. So when you think about gene editing or changing the genes in some way, of course, you start thinking about the aquabounty, which was then the traditionally developed with traditional transgenic methods, where the growth hormone was just inserted randomly in the genome. This was the technique at that time, and that results in increased growth. Now with gene editing, you can just then knock out one gene in fish and then get an increased growth. Which is then exemplified here with the MSDN1 knockout in sea bream, which then results in increased flesh or double muscle. So that's a big difference in terms of precision, and also not adding a genetic material, which is of course a game changer also then for fish aquaculture. So, but also then in aquaculture, the only species that has been coming into use for gene editing are then knockout animals who target mutagenesis. But also there are cases where we insert genetic material, CRISPR, or just make SNP exchanges, but those haven't been come into use yet. So how does then gene editing in farmed fish differ from other farmed animals? Yeah, one of the big differences, of course, you have external fertilization, external development, and this makes it easier, of course, to edit, because you can inject directly outside the animal. And also fish has a very high fecundity, maybe compared to other animals, so you can really upscale the production of an edit quite quick. But one of the drawbacks is that because teleosts had early on in their evolution of teleosts, you had the duplication of the genome, and then you had other strains of salmon or other strains of fish also had this duplication, so you have often duplicated genes. So that's a little bit of a challenge working with functional genomics in fish species. And then you have the challenge that this is a water living animals, and of course they can escape, and you don't have the same control maybe as you have on land living animals, and that's extra challenge when it comes to regulation. And another challenge is often these fish species have very long generation time, so to introduce an edit into a strain takes quite a long time. So the traditional method then for using to introduce edits in fish is to then inject directly into the early development, and this is just shown from our lab where we CRISPR directly into the egg, and then you can, in this case, we knocked out the albino gene, so it's very efficient. But we also work with, or people are also working with alternative to this type of introduction of edits, like sperm electroporation, or working with cell lines and surrogacy. So cell lines is not so well developed for aquaculture species, but it's very interested in terms of vaccine development, because a lot of the epitopes cannot now be developed because there are no cell lines. So CRISPR is a tool that should be introduced in these two areas. And also you can also introduce edits then into germ cell culture. And then in this context, sort of take out germ cells from superior breeds, make the editing outside the animal, and then put them back into sterile hosts. So there are several ways to introduce edits into fish, but the most common one is an injection into newly fertilized embryos. And then also one area is very, very interesting, of course, is to edit for disease resistance. This can be knockout animal, but often when it comes to resistance, you need to introduce genetic elements. And an idea then, and hasn't been done yet, at least published, to put in, to move maybe a resistant gene from one closely related species or a strain of the same species into the same breed or a closely related species. So when you summarize the areas of interest for aquaculture is, of course, production trait, like we saw the MSD1, fish which grow faster. Also reproduction, where you have what we are working with, sterility, maturation, and breeding. For disease resistance, this is early for edits using fish, but it's a very high interest to have that to reduce the number of drugs and so on that's used in aquaculture facilities. And then new cell lines will be a really good target also for edits to be able to produce better vaccines. And the other thing is also change the genetics of the fish so that it can eat locally grown or logotrophic level feeds. So I just had a look over sort of what's going on and what's been published up until 2023, and it's mostly 4 different traits. So I just show you growth, reproduction, disease resistant, and meat quality. Where for growth, there are 2 now commercial species in aquaculture, which is then the double-muscled sea bream, which is the MSDN knockout, and the leptin receptor knockout in the tiger pufferfish, that are both produced in Japan and not considered GMO according to regulation. For reproductive traits, there are a number of studies, including sterility, and that's sterility both for having the fish not integration into wild population if escape, but also to reduce puberty, which is a problem. And then also it has been studied with sex determination, because you want to produce all female or all male populations of fish, because for some species it's very good to have the all female populations to reduce the problems of early sexual maturation in males, for example, and it can also be the reverse. So any models that can help with that are good. And then disease resistance, of course, a very interesting area, but here it's early on, and only there's a few strains that has been developed, which then has affected the innate immunity. And for meat quality, there's a few studies that has been trying to affect the omega-3 synthesis in the fillet, so you can get fish which produce more omega-3, although maybe living on vegetable feed, for example. So The two approved and known one as used in aquaculture now in Japan are this red zebra and tiger pufferfish. They both have a higher growth rate. But interestingly, these are then not allowed to be farmed in sea cages. They are farmed on land. And so that's a little bit of on the contrary, what we have been trying to do in the last few years then is maybe to develop the edited fish, which will be reared in sea cages, and up on the scale, this fish will be sterile and then not interbreast with wild salmon. So, and so far, I don't think there have been any genetically modified fish or animal that has been released into the environment like this. So if you want to do this, you have to consider the regulatory landscape, which is different for fish all throughout the world, and especially then in Europe, it's very strictly regulated, and also Norway belongs sort of to that regulation. So what we wanted to do was to apply to put a research trial on this sterile genetic fish, although we are in this very difficult regulatory system. And the reason we want to do this, of course, we have then developed this. We've been working on sterile fish for a long time, and now we have a model that we think we can produce 100% sterility, but we're not there yet. So, but we want to see how this fish will do in the sea cage, because previously it has been used triplet salmon in aquaculture in Norway, which are then sterile, although what they discovered when we were working on those and they put into sea cages in large scale, they got a lot of deformity and problems, which was a little bit unexpected. So we want to know when we develop this sterile genetic fish that they won't develop anything unexpected. And we have already working on just the sterility models for quite some time in salmon, where we work on two traits, both to inhibit germ cell formation, which I'm going to show today, and also then to has model to prevent maturation. And we have both models that we work with, but I'm going to show you for the case where we wanted to apply to put this sterile fish in sea cages. We are working on the inhibit germ cell formation model, which is essentially dead end knockout fish. And if the fish lacks dead end, they don't form germ cells. And these are then 100% germ cell free. And we also studied them to see that they don't produce any sex steroids. So they are essentially what we have called a virgin fish. And so we have studied this in two large trials indoor facility. And we have seen that they are sterile and germ cell free, and they never become sexually mature. The only thing that differs between control and knockout is that they then don't produce sex steroids, and they also then develop smaller livers, which we think is a function of them not building up energy to form the gonad. But otherwise, for the other welfare markets, we haven't noticed any differences. But what we wanted to do in this trial was then to try to put a small scale experiment where we have half amount of knockout fish, half amount of wild type fish in a smaller experiment where we have a 12 by 12 meter net pen, and then we have 4 smaller net pens within, and then they're in a common garden. And then we will have a double netting on the top, double netting on the side, and extra netting on the side to prevent from bird attacks, so which can happen. So the fish that we used, and this is a little bit the problem we had with the fish, was that the generation time is very long. So to be able to produce these knockouts, we actually crossed out F0 crispant to make an F1 generation, where we had a combination of dead end knockouts, heterozygous fish, and wild type fish then, which were a result of that mosaic crossout. And of course, these fish then arrive from this. They are all having mutation at the target site, but they have different types of mutations. But when you look at the fish that we were going to use, they all had sort of different mutation at the site, but they all resulted in reading frame shift. They were even making a stop codon at one site in the protein or another. And both these we have shown that they result in sterility, and the target site is in the RM mining domain of the dead end protein. So we're 100% sure that even though they have different types of mutated target sites, they will all not produce a functional dead end protein. So we had this experiment where we then had half amount of knockout fish, half amount of wild type fish. They were put in common garden, and we were planning them to put them then, rear them to harvest at sea cages just to see if there were anything unexpected that happened to them. So to do this in Norway, you have to then send application to the Norwegian Environmental Agency. Also, you have to send a short orientation to EU. Then the Norwegian Environmental Agency sends your application onwards to the scientific committee who makes the GMO risk assessment. And also they have another consideration, which is the sustainability social benefit, the ethics committee, Biotechnologies advisory board. And both these committees then gives the feedback back to the environmental agency, which gives the feedback back to us. So this is supposed to take 120 working days. And interesting, nobody ever used this regulation before, so we didn't know where to start, and it was a quite old regulation that was 30 years old. But we managed to fill out all the questions about the health and the environment and all these things that are supposedly to put in there. When we sent in the application last spring, and it took us many rounds with the scientific board to be able to make a complete evaluation of the experiment. And then we got the 70 pages assessment after 6 months. And they said that this, the VKM and the scientific committee said that they meant that it was insufficient proof that sterility, that we had sterility in all the fish, and also that the fertile controls could contain mutations in the dead end gene that could be then transferred to wild salmon strains. So we were a little bit frustrated with that, but it's actually theoretically possible that the wild type control can have a larger mutation in the area we assayed. So we sent a complaint and said that we can of course correct this, and we can also show one more time, like we had promised, to show that these fish were truly knockout fish. So we then exchanged all the siblings, wild type siblings, with wild type control. We provided double genotyping of all the fish, and this fish amount had reduced because we had some problem with multiplication because the fish were kept too long on land, so it was a little bit stressed, but we still had enough fish to run an experiment. And then we did have to sample all the different genotypes, not considering that they were knockout, we had to have all the genotype, and there was another 60 fish that we had to take out and show that the fish were completely sterile with histology and qPCR. So quite extensive work. But then it took us like 3 months to do that, and then they evaluated the application for another 2 months, and then they said, even though we had double genotyping and we had done all this extra work, they still thought that it could be sterility alleles in the little population of fish experiment, so that we couldn't put out these fish and we were not approved. So that was a little bit disappointing, and we didn't send a complaint because now the fish, this was this summer, and now the fish is around 3 kilos, so it wouldn't have been useful to use this experiment. So we are now planning to reuse this sterile fish instead for some other experiments, like surrogacy studies, where we try to put in germ cells into this big fish to see what we can short generation time. We also will do what they required us to do. They wanted to see if these sterile fish actually eat more small salmon than other salmon. So salmon don't really eat each other very much, but if they're hungry, I guess they can eat each other. So then we will have an experiment where we take this big fish and it's a small fish. And then we're also going to reapply for a sea cage trial using the F2 generation, where we can have only one genotype then. So I think that could be the key to be able to get to do that. And otherwise, we will continue to work with developing breeding for 100% sterility, which looks really promising. So to conclude, I mean, aquaculture is experiencing intensive growth. There are over 20,000 species of fish. And gene editing is for sure a new toolbox in breeding, vaccine development, and general production. And it's been very hard to challenge this old regulation, I must say. I think both for regulators and for us, because we didn't really know how to approach, and we're now happy that we sort of know what's required. So I think by adding some new data and so on and new lines, we can probably, hopefully get through this complicated system. But it requires, this regulatory work requires 100% more effort compared to ordinary science, that's for sure. And of course, in retrospective, it would have been better to wait for F2 generation rather than to apply for F1 because it was too complicated. And it's strange the current risk assessment do not consider the benefits of the solution, such as saving wild salmon rate and the control fish, which has then 100% capability to spread farm genes into environment. How can that be allowed when this is not allowed? That's a little bit unclear to us working with it. But in the end, I just wanted to acknowledge my group here in Bergen for all the work that we've been doing on the sterility, and the collaborator University Utrecht, Syntec, and the industry partner Aqua and Greg. And I also want to send thank you to the authorities, which has been also, it's been a lot of work for them to try to assay this application that we have done. So especially the Norwegian Environmental Agency. And then in the end, I want to thank the funders. So thank you.